Hello everyone, my name is Stephen Calciano, and I'm the SCIEX Biopharmaceutical Market Development Manager for Capillary Electrophoresis in the Americas. I'm happy to introduce our speaker, Peter Holper, who obtained his degree in Biochemistry and Cell Biology from the University of California, San Diego. Peter has over 12 years of experience in biopharma, including as an analytical chemist at the Eli Lilly and Company, where he was responsible for developing the analytical control strategy for bioproducts. Peter currently works at SCIEX in Redwood City, California, where he is responsible for developing and optimizing CE applications and providing customer demo sample analysis support. Thank you so much for joining us today, Peter, and I look forward to your presentation.
Thank you very much for the introduction. As mentioned, I have a long history as an analytical development chemist who, like probably most of you are now, was always searching for ways to improve my workflow, really improve the methods that I had. And at the end of the day, I was always looking for a better understanding of the analytes that I'm working with, so that I could always be one step ahead of the questions that our regulatory agencies would ultimately ask us. Well, I'm excited to be here, to be able to share a potentially new way to characterize residual nucleic acids. So we'll start today with a brief background introduction to those residual nucleic acids, where they're coming from, why we characterize them. Then we'll look at ways we're currently performing this type of characterization. And from there, we'll dive into how we can really improve upon those workflows to give ourselves and the agencies that are in charge of approving our BLAs a better understanding of what is in our products. Let's start with looking into where these residual nucleic acids come from, right? Our cell culture processes. Now, the bioproducts we manufacture and we hope to bring to market are produced using cell culture platforms. And in recent years, new platforms have become really increasingly attractive to our manufacturing teams. They're based on production systems that can provide really a number of different advantages, such as a broad selection of candidate cell lines for product development, the possibility of more stringent product control and better product safety, or even ease of scaling up your production, and potentially reduce manufacturing costs. But there are always going to be safety concerns that are specific to these cell culture-derived vaccines and biologics. And these need to be addressed before regulatory approval of the products can be made. And many of these issues stem from process residuals. These are all derived from the cell substrates that remain in our final products as impurities. Now, the guidelines that define acceptable levels of process residuals in biologics, really by various regulatory agencies, have evolved over the last few decades. We can look all the way back to 1987, when the World Health Organization was aware of the risk associated with residual DNA. And they set a limit of less than or equal to 100 picograms in a single parenteral dose. Well, that limit was really intended to minimize the potential oncogenic risk of residual DNA from products derived from these continuous cell lines that we use. But subsequent information suggested that the residual DNA from these cell lines had a much lower risk than they really assumed, and they should just be considered a general impurity. Following that thought, in 1998, the limit was increased to less than or equal to 10 nanogram per dose. Then in 2001, the FDA recommended that size should also be taken into account when evaluating residual DNA. And in 2010, they followed up that with the recommendation that DNA fragments should be reduced to below the size of a functional gene, roughly about 200 base pairs. Now today, the regulatory bodies really agree on taking a risk assessment approach where we're taking into account the intended use of the product along with the size, the quantity, and even the activity of those residual fragments. Really, this is a case-by-case risk-based approach that they've advocated in recognition that residual DNA in some products might be higher than 10 nanogram per dose, and it might be very difficult to reach that 10 nanogram per dose level without impairing the potency of the product. And in our gene therapy space, where we have a lot of cell-based therapies and gene-based therapies, when we have lower titers, that becomes even harder to reach, a harder threshold to reach. So how are we meeting these characterizations need today, these needs today? And how can we do better? Well, one approach we use is just quantitation by PicoGreen. We have a dye that binds to double-stranded DNA and generates a fluorescent signal, whereas unbound dye emits very little fluorescent signal. Pretty simple. One of the advantages, right, is it's very simple to use, very low cost, and it has some utility for samples with high levels of DNA that you might see in your upstream processes. But PicoGreen has relatively poor sensitivity and can really only detect about 0.2 nanograms of a pure solution of DNA. Like all methods for residuals, it's also prone to interference from other components, and it doesn't always give reliable results unless you've very well extracted it. But the lack of sensitivity might be its most major limitation, especially given the guidance from the WHO to where you really want to set your threshold at about 10 nanogram per dose. And therefore, we generally don't want to use this for final product release, especially when we're using high dose products. Another technique that's used for residual DNA quantitation is hybridization. And here we're using probes designed to recognize specific target single-stranded sequences that are labeled with either a radioactive tag or a fluorescent dye, and we hybridize it with denatured DNA samples immobilized on a membrane. The amount of DNA is estimated from the signal intensity relative to a standard, which is generally the same genomic DNA from the host. And the advantages here is that it's similar to a southern blot, which is widely used among our peers. And so there's really no special training needed, and the cost is relatively low. And this can also estimate the size of DNA, because you can electrophorese the DNA prior to blotting across your standards of different size range. So like PicoGreen though, one of the major drawbacks is another lack of sensitivity, usually right about 10 nanogram per mil. It also has a pretty long testing time, up to upwards of 2 days. And that's really why we've steered away from this method in more recent years. Threshold is another assay that's been used. And here we have a single-stranded DNA binding protein that's used to bind and capture denatured single-stranded residual DNA, and a urease conjugated anti-DNA antibody is used to detect the bound DNA. And then by hydrolyzing that urea by an enzyme, we'll produce a change in pH, and the rate of change in that pH is proportional to the amount of DNA in the sample, which can then be calculated relative to a signal on a standard. This method is very sensitive, getting down to the 1 to 3 picogram range, and it has a very standardized protocol. But the throughput is low. That doesn't help us, and the cost is high. Another point that we have to consider here is just the limitation of the detectable size of DNA fragments. Because it depends on binding of a denatured DNA to a single-stranded DNA binding protein and the anti-DNA antibody, DNA fragments lower than about 600 base pairs are going to be missed. And during bioprocessing, it's certainly possible that a DNA strand will be sheared to smaller sizes during treatment, right? Whether through filters or purification columns. And we really need to know that size. That size is becoming increasingly important to better characterize our product. Now, PCR-based technology can also be used for the determination of residual DNA and is now easily the most widely used method in our industry. During the PCR reaction, a sequence-specific DNA template is amplified to produce billions of copies within a couple hours. And that allows for the detection of extremely low levels of DNA. Now, PCR products are going to be measured in the early stage of amplification when we're using real-time PCR, which allows for an accurate quantitation of the residual DNA in our products, because there's a high correlation between the level of DNA, of starting DNA, and the signals detected in that early exponential phase. Now, you can use several different chemistries, right? 3 very common ones include CyberGreen or dye-based primer signaling and TACMan. But as with most assays that we've come to love and enjoy, we know that speed doesn't equal precision, which is one of the major disadvantages here. And this assay in particular doesn't have the ability to provide size determination or the ability really to detect fragments that don't contain the target sequence. So here again, you're missing a fair amount of crucial information. So on the whole, these 4 technologies have a variety of strengths. Some are really fast, some are very low cost, some have very simple methodologies. But none of them really combines all of them. And most don't provide size characterization, which is really important in the eyes of our regulatory bodies. And in fact, I think we can sum up the shortcomings in 3 ways. Okay? They have inadequate sensitivity, they have insufficient selectivity, and they have too much rigidity. Where can we go from here? We really have to broaden our search to new technology that can rise to the challenge. Think beyond where we're used to playing. And that search can bring us to tools commonly used in analytical development that we already have in our labs. So we turn to capillary electrophoresis to better characterize our process residuals. Now, CE can provide a convenient method for nucleic acid characterization. This type of characterization has been done on our platform for really decades, and it has dedicated kit-based solutions already made. The SCIEX double-stranded DNA 1000 kit is one of those kits that's suitable, really, for rapid and automated analysis of these products. It has a replaceable gel buffer and a coated capillary that gives us really good reproducibility, which will then allow us to quickly characterize our products during process development, during manufacturing, during release. All very important phases of the product life cycle. Now let's look at this kit in action. Right, so on the top left of our screen, you can see that this kit can provide baseline resolution of 11 restriction enzyme fragments of bacteriophage DNA. And their estimated lengths are listed there. Well, the separation was performed with just a 30 centimeter effective length DNA-coated capillary, that same double-stranded DNA 1000 gel, and LiftFluor enhanced CE dye. The 2 red arrows that you see really just indicate deceiving potential. We see 2 fragments with a 10 base pair size difference, baseline resolved. On the bottom left, we see an example of a successful separation and sizing of restriction fragments of virus and plasmid DNA using the same kit. Peaks in green font are from a Hay3 restriction fragments of a bacteriophage DNA. And then a linear plasmid DNA is labeled in blue font. And then fragments from a 1 kb plus DNA ladder are labeled in black font. It provides very strong separation. But another aspect, very important aspect of residual characterization is sensitivity. Because without sufficient sensitivity, we can't prove that we're at or below the threshold of 10 nanogram per dose. So here we see serial dilution of the same 11 restriction fragments of bacteriophage DNA. And using the same Liflor enhanced C stain, we can obtain a LOD below 1 nanogram per mil and an LOQ of just under 2 nanogram per mil. Pretty sensitive. But I know, just like you know, that we always have people out there pushing the limits of instrumentation, just like I did for many years. And this application that we're showing today is really no different. Well, the scientists at MedImmune, now a member of AstraZeneca, were one set of those users that did that. They really pushed the limits of this type of assay. They explored ways to increase sensitivity by focusing on using different dyes, optimizing their sample matrices, optimizing their injection parameters, and even looking at the dye concentration to reach what was previously unheard of levels of sensitivity. Because they were able to achieve an LOQ of 0.025 nanogram per mil using the standard. And they were even able to identify the same 10 ladder peaks at 0.00625 nanogram per mil. And that is amazingly sensitive. That is great work by that team. And by following some of those parameters, not all of them, I was able to easily achieve sensitive and reproducible separations well below that 1 nanogram per mil range. And I understood that had I followed everything they were doing, I would have likely achieved even far greater sensitivity that they were able to. But how can we put that great work to work for us? Right? Luckily, we don't have to rely on analyzing a full ladder and comparing it to each sample we want to analyze when we want to characterize our residuals. We can really zero in on specific DNA fragments that are most important. And here, as we've been talking about, following our regulatory guidance, that 200 base pair fragment is really the threshold where we want to be able to accurately measure. So we need to have the sensitivity sort of to see this line in the sand, in this case, the peak in the electrophoregram, and how that compares to our samples. So we can analyze the 200 base pair DNA fragment by itself, and we can compare it to residual DNA samples that we analyze. That's certainly better than running a whole ladder standard, and it's fine. That's a fine solution. We have robust migration times. We can get very sensitive method. But we should always push ourselves to do better. Because if we don't, well, someone else is going to do it first. So what else can we do? So why not just spike the marker into the sample itself? It can give us a clear marker of that 200 base pair threshold we want to be under. ideally. And it allows for really the simplest of integration because you have a well-defined peak right there at 200 base pair. And we have complete confidence in the results we obtain. So here we are. We're looking at a residual DNA sample with a 200 base pair marker spiked in at a concentration of about 1 nanogram per mil. And you can easily estimate the total DNA within the electrophoregram if you'd like. We have linear response that we've shown earlier. Or you can simply rely on really the electrophoregram's ability to characterize your size of DNA if you quantitate it by another method. But in any case, you get a better picture. You get a more complete picture. You get better characterization. You get more confidence in the product you're hoping to bring to market. And in the continually evolving gene therapy, cell therapy space, really in all of biopharma, this is what you're going to need right now. This is what you're going to be pushed to in the very near future. Now we move to residual RNA. Can we take what we've learned analyzing that residual DNA and put it to use when we need to characterize residual RNA? Of course. But why RNA? Why are we bothering? Well, certain expression systems can leave trace amounts of residual RNA. One of the more commonly ones that's used is E. coli. And that can certainly be harnessed to express insulins, proteins, plasmids. Plasmids really are incredibly important by themselves. Now, how important? Well, really incredibly important to our therapeutics. They can be used by themselves as vaccines. They can be used as a gene delivery vehicle. They can be used to make process intermediates for our viral vectors, such as AAVs and lentiviruses. Truly important. And with all this regulatory focus on residual DNA, why are we worrying about RNA? Well, to be honest, right, we need to be worried about any impurity we have in our sample, in our processes. And it needs to be minimized and controlled, and we need to prove to our regulatory bodies that we have control over it, that we can see it if it's there. Because RNA in particular can turn genes on and off. It can alter other molecules and reactions. It can even build proteins. Right? So we should all want to minimize the introduction of any host cell products into our patients that we're looking to help. RNA is certainly no exception to the rule. Now we can easily analyze plasmids using CE technology, using that same double-stranded DNA 1000 kit actually. And this has been done for many, many years to monitor the different isoforms. But to characterize RNA, we need to take a slightly different approach. And in fact, we have a whole different kit just for that. Our single-stranded DNA 100R kit really harnesses the same capillary gel electrophoresis technology as our double-stranded DNA 1000 kit. But here we use a denaturant to limit the secondary structure of RNA to ensure a more accurate size estimation. And while this kit, out of the box, is really designed for rapid and reproducible separation between 100 or 10 and 100 bases, by simply diluting the sieving gel, you can get a much greater sieving range for your analyte that needs to be characterized. So it's really customizable to the size you're looking to monitor. And we have another solution that isn't currently a kit, but it's really optimized to separate up to about 5,000 or 6,000 bases. And in both cases, we have great sensitivity. Here we show an LOQ of about 0.3 nanogram per mil and an LOD of about 0.1, or really below 0.1 actually, nanogram per mil. And even here, we can take the learnings from our residual DNA analysis and put them to work to further increase its sensitivity. We optimize our dye concentration. We optimize our injection parameters. We find, by looking at some real-world samples, that RNA that's carried over from our various processes can have quite the size distribution. There really isn't necessarily going to be one main fragment for us to characterize. It can be spread out all over the place. We take what we learn and apply it. We increase our sensitivity. We characterize a sample against a size standard, or if we have it, if it's available, a single RNA fragment. And here in this sample as well, we can see quite a size distribution of RNA. Here we're looking at multiple products. See the size distribution in real samples. Each one needs to be monitored. Each one should be characterized to better understand how to minimize its presence. The final question that I wanted to explore and try to solve was whether we could monitor residual DNA in the presence of a plasmid. So we took that same plasmid that you saw a few slides ago. We also took some E. coli RNA, which you can see in the bottom left. And then the top left, you see the plasmid. Now, that's under denaturing conditions with a lot of changes to the method than you would in just analyzing that plasmid by itself for its isoforms. Then you spike in E. coli at just a 0.1% level relative to the plasmid concentration. We can see we can detect its presence, which is great. But we also see that the plasmid is likely co-migrating with at least some of the RNA that we wish to characterize. So I like to call this a work in progress. So what we have today, what we've been discussing, really is an approach to characterize residual host cell DNA by capillary gel electrophoresis with laser-induced fluorescence detection that allows for a great estimation of the size and the ability to quantify the amount. And with a few small changes to our gel, we can characterize RNA as well. And while additional sample preparation steps are probably required to eliminate interference from a plasmid, depending on its size, I think we have a really good starting point to continue developing that approach. And with that, I would like to thank everyone for taking their time today to listen to me. And would like to turn this back over to Stephen for the question and answer session.
Thank you, Peter, for these great insights into a high-performance residual nucleic acid characterization workflow. We're now going to move over to the Q&A portion of the webinar. But before we do, we'd like to pose a question to the audience. So what are your biggest challenges related to residual nucleic acid analysis workflows? You'll see a few answers that you can select from on the screen. Fantastic. Well, thank you, everyone, for your feedback. And we'll look to move over to the question and answer portion of the session. So please remember to post your questions to Peter by using the Ask a Question box on the left-hand side of your screen. So it looks like we have got a few coming in. And we will start with this first question. So are these low sensitivities exclusive to lift detection? And what can be achieved with PDA detection?
Yes, so everything that we showed here was using laser-induced fluorescence detection, using a few different dyes depending on the application, whether it was RNA or DNA. You can't use the PDA detector with this capillary because it is a coated capillary, and the PDA will actually burn off the coating, but you can use the UV detector. It's going to be more or less the same. It just has a single wavelength bandpass filter before the light hits the capillary window. And I guess in general, I would say the LIF affords you at least 10 times greater sensitivity, but I think that can be a little tricky here because these dyes are so powerful, really, compared to some of the other ones on the market, that it's probably even beyond that, even beyond 10 times, but I don't have an exact number off the top of my head here, because we can use a lot of the same tricks that we did to increase the sensitivity, that, how we're injecting the sample, et cetera, some of those modifications, but the enhanced sensitivity of the lift gets us really pushes it down to that sub-nanogram per mil level.
Fantastic. Thanks so much, Peter. I guess while we're on the topic, can you expand on what dyes you were using for the results shown?
Sure. So let's see. For this, where we started was using our enhanced CE lift floor dye. So that is one that we sell through SCIEX that you can order. And that is pretty sensitive dye, but to really push it farther, that we saw here, this is using CyberGreen 1, which has is going to bind more specifically to just DNA, whereas the enhanced LIF4 dye will have, some of a less of it could bind more towards either one, towards DNA or RNA. And then when we're moving on to here, to the RNA analysis, that's where we're switching to CyberGreen 2. So CyberGreen 2 now is going to be have a higher binding efficiency to RNA or single-stranded DNA. But both of those do have a much higher quantum yield than enhanced LIF4 dye. So that's why we do that, to push, really even farther the limits of what we can go.
Great, thank you so much. We do have a question related to, how did you optimize the sample injection?
Well, the sample injection, there's a few ways. And at first I started with our normal injection parameters, I guess, which would be about 1,000 volts for a couple seconds, 2 to 4 seconds. And looking at the publication that I referenced from MedImmune, they went all the way up to 8,000 volts for about 20 seconds. So that's going to be the high end. So I check a few in between, maybe 5,000 volts for 10, 20 seconds, and just see how it all plays out. And can you really go even push it above that 8,000 20-second range? So there is definitely a diminishing returns, I would say. If you go too high or too long, your reproducibility kind of suffers. So I saw good reproducibility where at that 8,000 volt for 20-second range. Oh, I know some other of our customers are backed it down a little bit. But what you saw for most all of these results shown was using that 8,000 volts for 20 seconds.
Perfect. Thank you so much. We've got a workflow-related question. So this one is, assume detecting CHO DNA. Is the dsDNA ready-made kit able to target this host DNA? Or is there something the user has to do related to primers or any other reagents? So,
yeah, there are no primers that are involved. At best, you would, potentially need to extract it or concentrate it depending on what that residual DNA is. But if it's there, I mean, you see the electropherograms shown were from a host of different sources, different production types. But yeah, there's nothing, I almost say, there's nothing special about CHO DNA that is going to cause an issue here. So it really is just the dye itself intercalating between those nucleic acids and the DNA strand.
Fantastic, thank you. One additional question about, does the kit quantify free-floating DNA in the sample only, or can it detect fragments that are partially attached to a product? For example, partially encapsulated DNA by AAV capsids.
That's an interesting question. I guess I didn't really look at that. Certainly it can quantify free-floating DNA, because if the dye can get in there and bind to it, then that's what's going to fluoresce. I don't know if it could enter, well, they say partially encapsulated, so I'm not I'm not sure if that DNA is going to be still attached then to some part of the capsid or what. So that might be something that I have to go back and dig a little deeper into.
Sure, not a problem. Thank you very much. We have a question related to analysis time. So what is the shortest analysis time, including preconditioning and rinsing, for a single sample?
Conditioning for at the start of a run is probably in the 10 to 15 minute range. And then a sample analysis, that can vary a little bit depending on the length of DNA you're using or the concentration of gel polymer that you've chosen that is optimal for your sample. Because certainly you can use it right out of the box at just the normal reconstitution volume, and that will give you some level of polymer that gives you a certain sieving range. If you dilute that out further, you get a larger sieving range, or you'll be able to separate in a shorter amount of time because your DNA will be flowing through that polymer faster. But I would guess about 20 minutes is a fair estimate for a separation time. It might go up to 30 depending if you're looking at something really big and want to, or relatively big, I guess, and need to keep that resolution between the peaks. I think 20 minutes or so is probably a fair estimate for separation time.
Great, thank you very much. A somewhat related question, what are ways to extend the life of the capillary?
Yeah, so because we're using a coated capillary, you can see degradation over time, and that is something that a number of our customers have looked into optimizing. And I didn't have firsthand experience putting this to work, but I can share what they've shared with me. One customer has used methanol rinses, both in the conditioning and shutdown steps, to, I assume, to keep the dye, or just to help rinse the dye out. Another one found that a shutdown method with gel that doesn't contain the dye actually extended the life quite considerably. So there is, potentially some thought there that the dye resting in the capillary for extended periods of time is not ideal. But those are very simple tweaks that you can make to the shutdown method or to your buffer trace setup to just get some more lifetime out of that capillary and still get great results from every day, every analysis.
Perfect. Some great tips to share. We do have another workflow related question. So after extracting DNA, is there any special sample treatment or preparation before going for analysis on the CE?
Not particularly. What I would say is you want to be cognizant of the ionic strength that's left over in your sample. So in the MedImmune paper, they discussed going to a certain amount of TE, so of the tris EDTA buffer that they were extracting in, and they found an optimal range, which was, I can't remember off the top of my head. But I didn't do that. That was something that I could have explored, but because I didn't do all those well, really I didn't have all that much material to work with, I'll be honest. So doing a buffer exchange on a minimal amount of material isn't exactly easy. But that would be the only thing that I would say to watch out for, is just try to keep the level of ionic strength lower, because we are using electrokinetic injection and we are really pushing the limits of that. So the less salt in there, the less salt is injected.
Okay, understood. Great, thanks so much. One other question. If there is a large amount of residual DNA and the range of fragments is large, with many fragments up to 50,000 base pairs, would the CE system detect a smear, and what is the, I think it's the limit of size?
Ooh, that's an interesting one. So if there's a large amount of residual DNA, you certainly want to dilute it out to the point that you're not maxing out the detector. So that's the first thing. You also want to make sure that you have enough dye, or an optimal concentration of dye in the gel. So that was one of the things that we looked at, is what is the optimal concentration for low levels. And you can kind of see a curve where you hit an optimal point for low levels of trace DNA, but if you have higher amounts, that ratio of dye to gel might not be optimal anymore. So that's another thing you have to watch out for. But for size up to 50,000, I definitely haven't looked at that. We have done plasmids up about 20,000, and that's really just looking at dilutions of gel. So I don't know that you necessarily see a smear, but you probably wouldn't get as sharp of peaks as you do in the smaller range. Or it may just be that your resolution between peaks isn't as great. But that would be an interesting thing to look at, is what is the optimal polymer concentration for such a sample. I haven't seen a standard that goes up to that range, so it'd be a little hard for me to test. But that's where I would start, is just look at different dilutions of gel and different concentrations of dye in that gel.
Fantastic. Thank you so much. I think we may have had a few more just come in, so just give me 1 second. All right, so I think that these have already been addressed. So with that said, I think we're ready to wrap up the Q&A portion of the webinar here today. So Peter, thank you so much for your presentation, and a big thank you to the audience for attending today's webinar and staying interactive during the Q&A session. So thank you very much, everyone, and we look forward to connecting again in the future.